Okay, so for this audio lecture, we are going to be looking at an introduction to microbiology. This is covered in chapter 1 of your textbook. I've taken chapter 1 and divided it into 2 PowerPoints, 2 audio lectures. Keep in mind that the audio lecture will cover like all the slides of the PowerPoint. During class time, we'll emphasize really key concepts, which I want everyone to know. So again, the audio lecture is more to help you. So that if I don't talk about something in class, but you still had a question about it, you could learn about it. Maybe, you want to hear my approach on it, my understanding of it versus like what's in the textbook. Okay. So feel free to use this audio lecture as needed, and feel free to kind of fast forward to the portions or the slides that you want to hear about, or replay the concepts that were talked about in class, to reinforce that information. All right, so for microbiology, a good definition of microbiology is the study of life, of living things too small to be seen without magnification. Now, that's the best definition. It's not perfect. So just like with anything with biology, there's always exceptions. Two parts of this definition have exceptions. So the part about living things, so when we talk about microbiology, we're talking about microorganisms or microbes. Sometimes we call these germs, gross things. So a lot of times in microbiology, it's the things nobody else wants to really study because they're like, yuck. They can cause disease. They don't necessarily cause disease. But in the definition of having living things, microbiology does include viruses and other infectious agents that are non-living, right? So one of the key components of being a living thing is that you're able to self-replicate, self-reproduce. Viruses and other infectious non-cellular, acellular agents need to have a host in order to make copies of themselves. So they don't reproduce, they replicate, right? So living things reproduce, they have a cell and they make a copy of that cell. And we'll definitely be talking about those processes as the semester goes on. But again, to kind of make, to have a working definition of what is microbiology, we generally say the study of living things. Just know the exception there with the living things is it does include viruses and these acellular infectious agents also. The other part is that the working definition of microbiology has in there that it's too small to be seen without magnification, right? So you need a microscope in order to see it. And that's where in the lab, you're going to be using a light microscope pretty much every single week, because an individual microorganism, for the most part, is too small to be able to see without magnification, without a microscope. So it's too small to be seen with the naked eye. And that's the other kind of exception. So there are some microorganisms that fall under microbiology that actually are large enough that you don't need a microscope to see them. It might be at some stage of their development in their life cycle they would fall under there. And one of the areas that is the case is when we're talking about hemolyths or parasitic worms. So parasitic worms, those are quite large. So if you've ever had a pet, a child, or even yourself, and you've passed a parasitic worm, you do not need a magnification. You do not need a microscope in order to see it. Now, for most of those, if they're in like a cyst phase or like an egg phase, yes, you would need a microscope in order to do that, and that's one of the reasons why a veterinarian and even an MD, a human doctor, would go and take a fecal sample and look at that underneath the microscope. They would either do a fecal smear or they would do a fecal flotation and then look at that underneath the microscope. But the adult stage, you usually do not need a microscope in order to see. There are also some bacteria that are quite large that you would be able to see those if they were grown under the right condition. Those Most of those are coming from different environmental microbes, not necessarily like human infectious microbes. So again, you should know that definition, but you should know why. It's not actually a great definition for that. So as I was mentioning, like with microbes, they have a lot of different functions and purposes. So when we look at microbiology, there's lots, we'll talk about like the different fields of microbiology. So this is meant to be kind of an overview of a lot of different information about microorganisms. There is going to be more of a human focus, just because of the textbook we're using, the course objectives, and also generally the interest, the student body that takes this course. So again, we'll focus a little bit more on microbes and some of them that cause disease, which you can see at the bottom of the slide. But there's a lot of other microbes out there in this universe. So they're very diverse, and that's what we'll get into in this class, and also just in this audio lecture, is talking about all the different types of microorganisms that there are, and some of the diversity of those. They're literally found everywhere on the planet, so even places where we thought they did not exist, they exist. So one of the things we like to talk is about like sterile environments. Well, even sterile environments like are not perfect. So if you think about like NASA and when they're building the space shuttle, like that's supposed to be a sterile environment. But even there, sometimes there's microbes. They've done studies where these sterile environments aren't actually sterile. Okay. So again, everywhere on the planet, there are microbes that can live in those different niches, those different environments. So if we're going to study them in the lab, we need to replicate that environment. So if we're going to study extremophiles that live in really hot environments, we need to make sure that we're replicating that to study them in the lab. And sometimes we can't do that. So it's actually a very small percent of microbes that we can actually study in the lab. So we have to think about what techniques, and some of these techniques that have been developed in the last decade or so, that are used to study them. They play a major role in different ecosystems. So if we look at like agricultural purposes, they play a really important role in making sure they provide nutrients. They can also carry out photosynthesis. They can also undergo nitrogen fixation. So again, from an environmental standpoint, really important also. Kind of linking that to humans, they're really also important for us in our gut to make sure that we are getting the max amount of nutrients for our body to absorb. Keep in mind that they're doing that for themselves. Okay. And then there's also lots of different beneficial uses we use in our society. So, it'd be, if you look in your fridge, there is most likely some kind of food or beverage in your fridge that is thanks to microbes. So anything from chocolate to cheese, to bread, to beer, wine, spirits, hard liquor, all of those are thanks to microbes. Even things like molasses and vinegar, like industrial production of certain chemicals are thanks to microbes. We also, a lot of our antibiotics are derived from microbes. So what chemicals do microbes produce to compete against other microbes? So those have been isolated, and then we use those so that if people become sick, if we have a disease caused by a microorganism, the antibiotic would fight that. So a lot of our, there's been this new approach, or not really a new approach. We're going back to the old school way of finding antibiotics by looking at what are these chemicals that microbes are going to end up producing. And we'll, again, look at that later on. And again, as I mentioned, production of nutrients, but also vitamins is really important. So like, it's one of the reasons why there's such a push for like probiotics or prebiotics, making sure that your microbes are being taken care of so they can take care of you. So in your gut, most of us have E. coli. non-pathogenic E. coli that actually produces vitamin K for us. So, I always like to thank my microbes for doing that for me. So there's lots of good things microbes do. Unfortunately, a lot of times in our society, we focus on what they don't do well or what they do well that harms us, and they can cause disease. So not only in people, but plants and animals, other animals like veterinary medicine. Obviously, infectious diseases are of concern due to a wide range of different microbes. But keep in mind that each microbe has a niche that they like to live in. So really, it's a limited number of microbes that, it may not seem that way, but there's a limited number of microbes that can actually grow in you or on you, opposed to what is out there in the world. Okay. So as I mentioned, there's lots of different members to the microbial world, and so they fall into different categories. So some of these organisms are cellular organisms, but they're also acellular or non-cellular, which are not living, that also fall into the microbe category. But again, one of the big criteria that we use is it small? Do we need to use a microscope in order to see that? And again, there's always exceptions to that. And then we can look at different structures that are present, and what you're seeing here is kind of like a dichotomous key where you can see on the left-hand side there is this cellular group of microbes, which include fungi, protists, bacteria, archaea. Okay, so each of these have different characteristics. So if we were to look at them under the microscope, we could look at what structures we see, what characteristics it has, what are the chemical compositions of things like their plasma membrane to indicate what group it falls under. And as we'll talk about in a moment, we can also look at the ribosomes and the ribosomal RNA to indicate what category it falls under. One of the big distinctions in the cellular group is whether it's prokaryotic or eukaryotic. And I'm going to say, let's hold that, and we're going to talk about that in just a moment. On the right-hand side of this figure, you'll notice that there are the acellular, the ones that most students, are most familiar with coming into this class is viruses. And later on this semester, we'll cover the chapter on viruses and how they replicate, and a little bit about their kind of life cycle and the different types of viruses. Okay. But they're going to be made up of proteins and nucleic acids. We also have viroids, which are RNA based. So viruses, their nucleic acids can be DNA or RNA, and they can be single-stranded or double-stranded. Viroids are only RNA, and it's a very small amount, and they tend to be circular. We also have satellite, and again, this is going to be composed of nucleic acids also. And then we also have the purons. These purons are a protein-based infectious agent. And so these are a really interesting group. And so later on this semester, we'll talk more about purons and some of the puron diseases. So some of the puron diseases affect the central nervous system. And again, they're infectious agents, so the transmission of them is, again, very interesting. So they're one of my favorite groups of microbes, of the less studied microbes. So as I was mentioning, when we're talking about the cellular microbes, we can break them into two different categories, prokaryotic versus eukaryotic. And again, we can look at some of the components of them. And one of the biggest thing traditionally is that we talk about prokaryotes lacking compartments, lacking organelles. And I caution to say this because what increasing evidence is showing is that there are prokaryotic cells that actually do have some compartments. And again, we'll end up talking about those. And then with eukaryotic cells, they're very similar to you, right? We humans are eukaryotic, and so we have nucleuses and we have membrane-bound organelles that have special functions. And so these eukaryotic cells like algae, protozoa, hemolymph, and fungi have a lot of shared characteristics with us. But again, there's gonna be special things that they have, like algae is going to have chloroplast. Protozoas, they're single-celled. Parasitic worms, they don't have the developed organ systems like us, but they are multicellular, and they do have certain organ systems. And fungi can either be single-cell or multi-cell, depending upon whether they're mold or yeast. So yeast is single-celled fungi, and molds are multicellular fungi. And then there are the viruses, right? So again, as I mentioned, those are acellular, non-cellular. So they do not have a cell. So they are parasitic in that they have to infect a host. They'll have proteins that coat their nucleic acids, their genome. And again, they need to infect some host cell in order to make copies of themselves. Okay. And as I mentioned, looking at figure 1.1, there are additional non-cellular, acellular infectious agents, but the viruses are the largest kind of category that we will look at, and again, most students have an understanding of. So when we classify, one of the first questions when you're looking at a microorganism is what category does it fall under? Is it a prokaryote? It is a bacteria, archaea. And so bacteria we recognize as being true bacteria. Archaea tend to be more environmental in very extreme environments. There is some evidence showing that archaea play a role in like dental disease. So if you're going into the dental field, you might have to know about archaea, but otherwise, you may not come across archaea unless you decide to study, thermal vents in the sea or lava fields in like Hawaii, right? So archaea tend, again, to be more environmental. And then eukarya would be the eukaryotes. These next couple slides just show you some of the different characteristics between these different domains, between bacteria, archaea, and eukarya. So I'm not going to read through these, but and one of the reasons is there's this really nice table from your textbook that actually summarizes up some of the different properties, characteristics of bacteria versus archaea versus eukarya. So again, I never expect my students to memorize tables, but you should know some of these characteristics between these different domains, and as we cover them during this course. So in chapter 3, we'll be looking at more prokaryotic structures. In chapter 4, we'll be looking at eukaryotic structures. So I'm going to say stay tuned to that. But this is, again, when you're studying for the first exam, this is a nice table to kind of know some of this and think of like, how could I write this up in like 3 to 4 sentences for a short answer, the differences between the different microorganisms, or again, some of those components that we end up focusing on in class. And then again, don't forget the acellular infectious agents. And again, we're going to cover these in more detail at later on in the semester. And again, we'll spend most time on viruses, but viroids, satellites, and purons are very interesting also. All right, so the next kind of area that we're going to look at is kind of the history of these microorganisms and how they developed. So one of the things we want to keep in mind is if you look at this figure, you'll notice that humans are up here. So more recently, humans developed. If we start to look back on this timeline, what you're going to notice is more recently is where eukaryotic organisms came about. Before that, we had a pre-eukaryotic cell that developed a mitochondria in chloroplast, and again, we're going to talk about the theory behind how that happened. If we move back even further, that is where we can see the first cell that appeared. Okay, and that's what we're going to talk a little bit more about, is like this first cell that appeared and what were the characteristics of that cell. And then we're going to kind of like say, okay, well, from there, how do we move along? So what evidence has been shown is that the earliest molecule that occurred was RNA, right? So when we're thinking about nucleic acids, traditionally the dogma says that we go from DNA to RNA to protein. But again, historically, when they look at isolation of some of these early fossils, what they see are these RNA that are surrounded by lipids. So this liposome. And so that is believed to be the first early cell. So that RNA then was able to make protein, right? So we would have had to have like a ribosome in order to do that, in order to take those molecules, take amino acids, and then make proteins, right? And remember that the way evolution happens is when something has an added fitness, when it has that ability to survive, it gets to survive, right? So it's going to outcompete. So over time, then what happened was we ended up having a cell that had DNA, then RNA, and proteins, right? So DNA is more stable than RNA. So that would have given a cell that developed the ability to have DNA added fitness, right? So when we look at RNA for today's version versus these ancient versions that have been identified, one of the things we'll notice in cells and their RNA, in modern day cells, there are different forms of RNA, right, that end up associating with the ribosomes. So hopefully this is some intro bio coming back to you, maybe some genetics in there too, right? And so you're going to have ribosomal RNA, which is rRNA, transfer RNA, tRNA, and message RNA, mRNA, right? So again, these are going to end up associating your ribosomal RNA as part of the ribosome. It is going to be the ribosomal RNA bound with protein is what your ribosome is. And then with transfer RNA, that is going to bring that amino acid in so that you can then make the protein. So some of those, you can think of these as bells and whistles that we have in modern day cells, wouldn't have been there in the ancient versions, right? But over time, again, it would have given this added fitness, right? So that having that RNA seems to be a precursor to that developing double-stranded DNA, okay? And that the role, that importance of ATP we see being present. There are also some, if we're looking at these ancestral cells, there's the earliest metabolism that would have occurred would have been with inorganic compounds such as iron, sulfur. So these would have been really harsh environments, conditions that these cells were originally present, right? And so again, they would have had to been able to make use of these harsh inorganic compounds and been able to metabolize them and utilize them for energy source and such, okay? We also look at the process of photosynthesis. So photosynthesis, we can see cyan bacteria. So cyan bacteria, cyan blue. So these used to be called blue-green algae. They're actually bacteria. And so these actually date back to over 2.7 billion years ago. And they were able to undergo photosynthesis. And that, because they could undergo photosynthesis, that's why they got classified as algae, because we think of algae, you had to be eukaryotic to undergo photosynthesis. Guess what? You didn't. You could be bacteria. You could be a prokaryote, like cyanobacteria. It just wasn't housed in a membrane like the chloroplast. It's free chlorophyll. Chlorophyll is a pigment that can utilize light as an energy source to then undergo photosynthesis and produce glucose. So cyanobacteria would have had an added benefit of being able to produce its own glucose, right? And again, hold that because we'll end up talking more about those cyanobacteria and how did we get from these prokaryotic cells to these eukaryotic cells. And then you can see here, these are stromatolites. So these have mineralized layers of microorganisms. So again, this is where scientists can come in here and look at these microorganisms that have been kind of embedded into these layers and use things like carbon dating and such to look at what are these processes that were occurring in these organisms that are our ancient ancestors. And what they would have done is also looked at the ribosomal RNA. So they would have looked at the small subunit of the ribosomal RNA. And this is, if you're reading literature and you see SSU RNA, that's referring to that small subunit ribosomal RNA. And they will sequence that, and then they can align the ribosomal RNA sequences from different organisms to see where evolutionary they diverged, and you can end up creating these phylogenetic trees to again see how related something is to another organism. So what domain it's in, and again how closely that occurred. Using that technique, they've identified this last universal common ancestor, LUCA, and so what has been shown is that archaea in eukarya actually evolved independently of bacteria. So in some ways, we are more like archaea than we are like bacteria, right? So they diverged from the common ancestor, where traditionally theories were that archaea and bacteria would have had a common ancestor, right? So they're both prokaryotic cells, but that's not what data is actually showing. All right. And then, as I mentioned before, if we're looking at that timeline, we had that early cell that would have had RNA in it and then lipids around it to create this liposome. And then we had this pre-eukaryotic cell. So it wasn't like the eukaryotic cells we have nowadays, but it's like a precursor to that, right? And so I had said, we're going to end up talking about how we got from that pre-eukaryotic cell to the eukaryotic cells we have now. And if you were to identify a eukaryotic cell, so if I put an image on the exam and I said, is this a eukaryotic cell or is this a prokaryotic cell? One of the things you'd be looking for are these organelles, right? Organelles like a mitochondria, like chloroplast, right? So those are the two kind of intro bio, you would have been like, okay, what's the function of the mitochondria? What's the function of the chloroplast? And again, we'll end up talking about those when we cover chapter 4. So the question really becomes, how did these eukaryotic cells get a mitochondria in chloroplast? And that's where the endosymbiotic hypothesis tries to understand that process, right? And looking at the evidence to support this theory, right? And again, it tries to explain how some of the organelles arise in eukaryotic cells. And the important thing is to think about what are the functions, right? So what is the function of mitochondria? Well, mitochondria is going to produce ATP, hence why it's called the powerhouse of the cell. The chloroplast is going to undergo photosynthesis and produce glucose, right? There's also some really critical features of the mitochondria in the chloroplast. The mitochondria in the chloroplast have their own ribosomes. Those ribosomes are a different size than the ribosomes that are in the rest of the cell out in the cytoplasm or attached to the ER, in the rough ER. They also have their own genome. They have their own genes so they can produce enzymes which they need to provide their function. And so with those, in additional to looking at that ribosomal RNA and looking at those genes on the ribosomal RNA, so what is the sequence of it? Those genes of the ribosomal RNA, that sequence of the ribosomal RNA of the mitochondria in the chloroplast actually show that it comes from a bacterial lineage. Okay, so it doesn't come from a eukaryotic. Again, your eukaryotic ribosomal RNA has a different sequence than the ribosomal RNA of your mitochondria. Your mitochondrial ribosomal RNA sequence is very similar to bacteria. So it speaks to the mitochondria actually being from bacteria. And so what you're seeing here in this figure is that we had this protobacterium. Again, remember, the function of the mitochondria is to produce ATP. Prokaryotic cells produce ATP and they do that across their membrane. And they do that, they don't have mitochondria, right? And so it's what the hypothesis, the endosymbiotic hypothesis, that there was a symbiotic relationship between these protobacteria. So this cell had an added fitness because it can produce ATP because it had this bacteria inside, a symbiote. And that allowed it to survive. The protobacteria had a little protection from the outside world. So that was a nice environment. So it replicates and survives. Similarly with chloroplast, here we have our cyanobacteria. Cyanobacteria can undergo photosynthesis. What does chloroplast do? Undergo photosynthesis. And so what would have happened is that we would have had this cell that had a protobacterium in there that was now serving the function of the mitochondria. Now the cyanobacteria is taken up. Again, this gives this cell, this like pre-algae cell, the ability to undergo photosynthesis. So it's got glucose being made right there for it. It gives an added fitness. It's able to survive, right? So again, when we look at the endosymbiotic hypothesis, and again, this is going to come up when we look at eukaryotic structures in chapter 4, when we look at the ribosomal RNA, when we look at those ribosomes in the mitochondria in the chloroplast, when we look at the genome, right, those are very similar to protobacteria and cyanobacteria. So it speaks to that origin. There's also the hydrosomes. So hydrosomes is believed to be an anaerobic endosymbiote organism that, again, because it was being housed inside this pre-eukaryotic cell, it allowed that organism to survive, hence symbiote. It gave an added fitness to that cell that was housing it, so the cell was able to survive. If something's working, you continue to do it. So just like, hey, the RNA is not so stable, let's have double-stranded DNA that's more stable. Hey, we're not making ATP, but we need ATP, and for the cell to absorb ATP from its outside environment is not going to keep the cell alive. Hey, why don't we take that protobacterium in so we can survive? So again, one of the kind of key really to cell bio, to intro bio, any biology class is the endosymbiotic hypothesis. And again, sometimes it's referred to as a theory. We want to keep in mind that microbes evolve just like we think of, non-microbial organisms evolving also. So they can also have mutations to their genetic material that can lead to different traits. It could lead to the production of a toxin that gives it a survival. Production of a chemical like an antibiotic that allows it to outcompete its competition, right? So in some cases, new genes and genotypes can end up evolving. And that means that the colony, the microbes that are there, end up becoming this like mosaic. And this will be something that when we talk about antibiotic resistance, is important for us to acknowledge that with antibiotic resistance, a lot of time we're looking at a mosaic of maybe one species that's causing an infection, but some of them could carry the antibiotic resistance where some of them might. So you might start feeling better when you take the antibiotic, but that just actually selects, right? So that takes it from being a mosaic to just the ones that are resistant. So again, this idea of picking up virulence factors or having mutations to genes that give a survival, but there could be still a mosaic present is going to be something we talk about later on too. We'll also be talking about how genetic information is exchanged between the same species of microbes, but also to different species of microbes. And one of those things we'll talk about is horizontal gene transfer. And so this is something we see both in bacteria and archaea. And that increases genetic diversity among those organisms. All right, so looking at different microbial species, one of the things we'll end up talking about, this is a lot of we'll end up talking about how they reproduce can help us to identify what type of microorganism it is. So some eukaryotic microbes, they're going to actually undergo reproduction. They can undergo a asexual or asexual reproduction depending upon the microbe they are. Some of them can do both. So keep in mind with sexual reproduction that increases genetic diversity. So again, it's something we might see happening in eukaryotic microbes. So there are some protistas and hemolyphs that undergo sexual reproduction. But again, a lot of them will undergo asexual reproduction. With bacteria and archaea, they do not reproduce sexually, so they undergo asexual reproduction. And so when we're looking at a bacteria or archaea, we would identify the species of the organism, like genus and species. But if there's been enough divergence from that, we would also have strains, right? So sometimes there'll be 3 parts to the name. That third part indicates what strain it is. And sometimes that's named after who identified it. Other times that strain is the location it's at or some kind of special characteristics. And then you'll notice a number of the, on the slide, you'll notice these 4 terms, bioval, morphoval, seroval, and pathoval. We'll end up talking more about these later on in the semester. I just want to point out the last 2, seroval, that is where the strain is divergent enough that antibodies that were produced recognize it. So if we wanted to know the serotype of the infection, we would have specific antibodies for that strain and we would be able to identify them. They're different enough. It's where these antibodies are present. And then pathoval, that is where one would be pathogenic where the other one's not pathogenic. Okay. And then we want to keep in mind when we're naming the bacteria and archaea that we use a genus species. So just to give you an example, Staphylococcus aureus, or that would be abbreviated S. aureus. The genus name is always capitalized. The species is lowercase. Strain would also be lowercase. And again, you'll be working with a number of different species of bacteria in the lab. So you'll get very familiar with them and the different properties of them, what they look like underneath the microscope. And so a lot of times the name tells you about the organism. Sometimes they're just named after the scientist or where it was identified, but a lot of times they're named after characteristics. So Staphylococcus, these are clusters. So staph means cluster or grape-like. So it looks like a bunch of grapes. Coccus, spears. And then aureus is for sunset. So when the colonies are, when you have enough of the bacteria that you have a colony forming. that colony actually has like an orangey kind of peachy color to it, which is where the aureus comes from. Okay. Where other organisms like Micrococcus luteus light, it actually looks yellow when their colony forms. Micrococcus, that's actually these groups of 4. They're called tetrads in spears. Now, like Bacillus subtilis, that's going to be rod shaped. So it kind of looks like a hot dog underneath the microscope. And subtilis, I don't know where it got, just very common. So again, as you're working with these different bacteria under the microscope, you're going to end up doing staining, which we're going to actually talk about in this unit for chapter 2. We'll talk about some of the different stains that you'll end up doing during the semester that can help to identify what type of organisms you have, not only because of its shape and what we call arrangement, but also because of whether it's like gram positive or gram negative, or whether it's acid fast positive or acid fast negative. And again, there's other stains that can differentiate, just like there's media that can use to differentiate what type of organism you're working with. And again, as the semester goes on, I don't teach the labs here, but you're always more than welcome to ask me questions, as long as that's not going to violate, like, I can't give you the answers for the exam or any of yours. I don't know what the unknowns are at the end of the semester, so you can't ask me those questions. But if you have questions about some of the techniques, I did teach labs for a number of years, so I'm familiar with those techniques. And lastly, if you do have any questions of any of the information, please let me know if you have any questions. I'm more than happy to answer those.